Here we are going to demonstrate the shoot tip excision process. After the shoots have elongated from the nodes on the pretreatment medium, after about 2 or 3 weeks, they are ready for shoot tip excision. Here we are preparing our microscope with a sterile surface. Here we have a glass plate that has been sterilized that is on the microscope stage. And we're taking each segment that has elongated, putting it under the microscope. Here we have a closer look. The leaves are easily broken if bent, so you can either bend them to break them off of the stem, or you can cut them with a sharp scalpel blade. We're going to remove these little clear scales that are covering the shoot tip area so that we can get a better look. So here we have the shoot tip with 1 or 2 leaf primordia exposed. We're going to remove the other scale on the other side of the node. And now we are ready for the final cut. Using small micro blades for this is very helpful, and you get a little bit more of a precise cut. So just below the shoot tip area, you want to make that final cut. And here we have the finished shoot tip. At this point, they're going to be plated onto the pre-culture medium and incubated for 3 days at 25 degrees in the dark. Here we have another example of a different cultivar. Every species and cultivar is going to be a little bit different with some variation. Some may be a little bit more hairy than others. Some may be more tender than others. So you will just have to adapt to each cultivar or species depending on their unique traits. Here again, we're removing that clear scale that is covering the shoot tip area so we can get a better look at it. There we can see the shoot tip with a few leaf primordia covering it. And here we're bending the leaf, and again it breaks very easily. So that's usually the easiest way to get the leaves off, the bigger leaves. Now we're removing the other scale on the other side of the node. And we are now ready for the final cut just below the shoot tip area. And there we have our finished shoot tip. Should be about 1 millimeter tall and perhaps less in width, maybe about 0.5 millimeter in width.